Okay, so good afternoon. So today I would like to really kind of frame everything that I've been talking about and really kind of put it into context and explain really of this course, really why is it about the chemistry that explains the biology? And what we've covered in this course, because of course we started with the stuff to do with pH and forces, and then that came very nicely into talking about amino acids. But also pH and the intermolecular forces, they also have an element in lipids, carbohydrates, and I'll put it over here for spacing, nucleotides. And so it's also kind of involved in all of them because the intermolecular forces, they really explain why the carbohydrates they prefer water. They're water soluble, and they also have the water soluble, and also they have a structural role. Doesn't really explain too much that I'll go on to later when talking about metabolism, but that's a kind of to start it out a little bit. And then, but it does have more of a role in lipids because that's going to talk about how the lipids come together and how they, with the hydrophobic forces, and how you can create two different compartments with which are separated by a lipid, because most things in biology are separated, are water soluble and separated by lipids. And the hydrophobic effects can help to explain that. Amino acids, of course, because you have all of the different amino acids with all of the different R groups, which kind of controls their physical properties. And nucleotides, because of the way that nucleotides come together with the nucleic acids. And to the storage of information, and kind of like the two strands coming together. But the main thing really is, we were talking about amino acids, and let me change it then. Amino acids goes down into proteins, of course. And then proteins, we've got the function. And for the function, we talked about two different functions. We talked about functioning as kind of like a carrier, in terms of the hemoglobins, the example we had. But also we talked about enzymes as well. And enzymes catalyze chemical reactions. And this really is where it all comes in together. Because these enzymes, they help facilitate the lipids and the carbohydrates kind of coming together to produce energy. And this is where we're going to go with last of all. We're going to talk about energy. We're going to talk about how the body produces energy. Because the thing is that energy is required in order to keep things together. The second law of thermodynamics is that everything is going to go towards chaos, and that you're required to put in energy to stop that chaos. And our bodies are tightly formed, and they have a shape and a structure. But to keep that shape and structure requires energy. And if we were to lose the energy, we'd no longer have shape and structure. Such as, for instance, with the, when we die, we will lose our shape and structure, and we will rot and become CO2 and ammonia and water. And the same kind of thing. And those are those small, simple molecules that we will decay into when we die, because we no longer produce energy. But we are made out of larger, more complicated molecules. What's that? Nothing. So that's something which needs to be, that requires energy, and it's dictated by the second law of thermodynamics. And that energy is really breaking down lipids and carbohydrates in particular. It's also breaking down amino acids using enzymes, and those enzymes produce energy, and that energy will help you to build new carbohydrates, new lipids, new amino acids and proteins, and nucleotides, which are the structural part. So it's all kind of like links in together. We study the smaller molecules to study how they work. And at the end, I'm just going to give you just a little bit of a peek. It's largely really what we go into more of Biochemistry II, but just like a little bit of a peek of what, if we put all of this together and we use it to make energy, and the energy is going to keep everything together. And really, how does the chemistry explain that? So I'm going to give a presentation now. I'm going to go into that. And to frame this, kind of think of it as being that, let's start out with the complicated molecules. Carbohydrates, fats, and protein. They are quite complicated molecules. They're quite large, made up of lots of different parts, especially things like proteins. And they get broken down into simpler things. And when they get broken down into simpler things, you produce energy. You also produce entropy, which is chaos, because one carbohydrate will break down into multiple CO2s and multiple H2Os. And the way that I like to put it is that what is more chaotic than one toddler? Well, many toddlers. And so if you can kind of think of it as being carbohydrate is like an adult, and it breaks down into lots of children. Well, lots of children are a lot more chaotic than one adult. So as you increase the number of molecules, make them smaller and increase them, then you increase chaos, you increase entropy. Whereas if you put them together to make a small number of more complicated things, then you decrease chaos, you decrease entropy. So you break down these complex things, you make energy, you don't make energy, but you add energy to the biological system, and you increase the entropy. And so, for instance, ADP and oxidized cofactors are reduced, and you get ATP and reduced cofactors. And so these things are getting oxidized, and these things are getting reduced. And then that reduction and that ATP is a storage of energy. And that energy is then used to take the simple things, such as the amino acids, fatty acids, nucleotides, monosaccharides. These are smaller monomers. You take that energy and you use it to build the complicated things from amino acids. You build the protein. And then thus you break down the ATP to ADP, you reduce, you oxidize the cofactors, and the cycle goes around. Because you're taking simple things, turning into complex things, that costs energy, and it decreases the entropy. And that's that second law of thermodynamics, that to go from something complex to something simple to something complex, you're decreasing the entropy, and thus you are and so thus that requires energy. So I see that there's a chat message. I'm just going to have a quick look at that is. You're not able to see the slides. Okay. Can you see it now? I guess it's when I stand in front of it. Does it look okay now? I can see it on my screen. Can you guys see it? Yes. I can see it on my screen. Is it showing up okay for you guys? I'm going to record it and put it up on YouTube as well. So if it's like a streaming quality issue, then maybe it will. I got a couple of thumbs up. It looks good. Okay, good. All right, I'm going to keep going then. So we can break it down, the life on this planet, we can break it down into a few different things. And life can roughly break down into two types of organisms, autotrophs and heterotrophs. Autotrophs are things like plants and also algae. They're photosynthetic life. They are able to take energy from the sun and use it to turn CO2 into complicated molecules such as sugars. And then those sugars can turn into other things. Heterotrophs, on the other hand, eat those sugars made by the photosynthetic autotrophs and then can break them down. So that you and I are heterotrophs, and plants and algae are going to be autotrophs, taking the energy from the sun. And we, as heterotrophs, steal that energy from the plants that originally came from the sun. So the photosynthetic autotrophs produce sugars that we consume that turn into CO2, that they turn up to turn into sugars again. And the cycle goes around. Now, in this kind of process of when we are making things and breaking them down, the way this is in between, these are metabolites. So metabolites are intermediates of turning one chemical into another in your body, in turning sugar, glucose into glucose 6-phosphate, into pyruvate. Those are all metabolites. And these metabolites together, they make up intermediary metabolism, which is all of the metabolic processes in the cell. You can kind of see it as being this big map that's arranged kind of like an underground system in a city. And if we were to just zoom in on one little section on this big complicated map, you'll see that it's just these pathways of glucose to glucose 6-phosphate to fructose 6-phosphate, catalyzed by enzymatic reactions. And there's this huge metro map that comes together to make that. And it's extremely complicated, actually. This is it kind of zoomed in. And this is just a part of it. This is just a tiny, tiny part of it. This is like 1% of the processes. So it's extremely, extremely complicated. But if we're able to zoom in on this, this is the intermediary metabolism. And these are all individual different metabolites that are linked, that make all of the things that make us us. And they're often made by enzymes, converting them from one thing into another. Okay, I've got a little bit of an audience now, so I don't have to talk at the computer. I can talk out. It's really distracting talking to the computer, so this makes it a lot easier. I appreciate that. So just a few terms as well, getting into physics, just a little bit. I'll start actually, so I've already defined entropy. Entropy broadly, a physicist is going to beat me over the head over it, but entropy broadly is a measure of chaos. And the chaos is normally like more things, more chaos, fewer things, less chaos. A small number of big things is less chaotic than a large number of small things. That's generally that. And a measure of organization. On one side is chaos, on the other side is organization. Catabolism is breaking things down. Anabolism is making things. Maybe you've heard of like Arnold Schwarzenegger, a bodybuilder, taking anabolic steroids. Anabolic means to build. So there's steroids which increase building of muscle. So anabolism is making things. Catabolism is breaking things. When you break things down, you break them into smaller parts. So the entropy goes up, but you get energy. On the other hand, anabolism, you make things from smaller parts, so it gets less chaotic, more organized, but it costs energy. And you can kind of see it here, where small things are turned into big things. It requires energy, but then that energy comes back by breaking down big things into small things again. And this really frames this big concept that I'm quite big on in terms of metabolic biochemistry, is that the catabolic pathways, such as the breakdown of lipids, breakdown of sugars, breakdown of amino acids, catabolic pathways tend to converge. And the thing is, if you think about all of the different molecules which you have in your body, all of the different fats, all of the different phospholipids, all of the different triglycerides, all of the different fatty acids, all of the different steroids, and I'm just talking about lipids. Then we've got the 20 different proteogenic amino acids, then other amino acids, then all of the different sugars. There's a huge list of all of the different metabolites present in life. And if you had just 20 different enzymes to break down each of those, you're going to run out, you're going to have a ton of enzymes. There's going to be a lot of redundancy too, because a lot of these are similar. Actually, the way that life does it is that it tends to converge. What I mean by that is that everything actually tends to produce acetyl-CoA or a TCA cycle intermediate. And so, for instance, phospholipids and triglycerides can be broken down into a common agent, and then that common agent can be turned into acetyl-CoA by beta oxidation, which is for another time and place. But for instance, amino acids, when we break down amino acids, several amino acids break down to pyruvate, and then they can join in and breaking down pyruvate to TCA cycle intermediates. So why bother having a way to go from glycine to some kind of energy source? Why don't we just convert glycine into pyruvate? Why don't we just convert alanine into pyruvate? Why don't we just convert aspartic acid into oxaloacetate? And then if we convert all into those similar things, it means that things overlap. And it makes it so that you can just reuse the same metabolic enzymes rather than having a different metabolic enzyme. So most metabolites in life tend to eventually converge when you break them down into a TCA cycle intermediate. On the other hand, that means that you can build out from TCA cycle intermediates. Anabolic pathways diverge. Anabolic pathways means you can build things from simpler things, but it tends to be the same simpler things. You can make amino acids from other amino acids. And if you start with a simple amino acid, you can turn it into a more complicated amino acid. Now, there's a lot more to it than that. And that's kind of only like 80% true. But the idea is true, that you can make kind of similar, simpler things and then turn them into the more complicated thing. Rather than having a different synthetic route for all of the 20 amino acids, why don't you just find a route to turn it into glycine, and then glycine into each of the other things. And then it just decreases the number of steps you have to do. So catabolic processes converge onto the TCA cycle. Anabolic pathways tend to pull out of the TCA cycle. This is another metabolic process. Honestly, I'm not going to go over this in too much detail. I'm not going to. This is kind of switching up gears a little bit, and I don't want to go into. I don't think we have the time. I don't want to go into a ton of detail about this, but I just wanted to mention that a lot of the time, some of these metabolic steps, they have kind of sub-steps that break down. For instance, in an enzymatic reaction, there will be steps in the mechanism, and some of those steps may be exergonic, and some of those steps may be endergonic. And really, what I want to say really is that sometimes if you have a chemical reaction which is unfavorable, you'll do a favorable one first to make the unfavorable one more likely to happen. And so if you need to go from A to C, you'll go via B because B will be an easier route. Let me explain what that means. Give a diagram. So for instance, let's say that you want to go from glucose to glucose 6-phosphate. And that is going to require energy, quite difficult to do. So what we do is that we team it up with two easier reactions. The first one is going to be the breakdown of ATP to ADP. And then the second one is putting that phosphate onto the glucose. And by teaming together unfavorable reaction with a favorable reaction, the net is a reaction that will proceed. So to go from glucose to glucose 6-phosphate, that's just kind of one metabolic path. But we'll break it down into two sub-steps because to go from glucose to glucose 6-phosphate is difficult. But if we it's endergonic, it requires energy. But if we couple it with an exergonic reaction that gives energy, such as the hydrolysis of ATP to ADP, that hydrolysis releases energy. And if we couple it to the energy requiring reaction, then that will make the overall reaction occur. So really, this is a really good way of framing why do we make ATP? Well, I'm talking about making energy. I'm talking about metabolizing food and turning it into energy. What's the big deal? Why do we turn it into ATP? And the reason is that ATP is a really good source of chemical reaction, where if we hydrolyze ATP into ADP, it releases a ton of energy, about 30.5 kilojoules per mole of energy. It's quite a lot of energy. And so if we can team up the hydrolysis of ATP with the unfavorable reactions, this one releases 30.5 kilojoules. That's the negative number, means it's releasing. This requires energy, 13.8 kilojoules per mole. The phosphorylation requires 13.8. The dephosphorylation of ATP releases 30.5. So overall, you add those up together, 13.8 kilojoules per mole required, 30.5 kilojoules per mole released, which means that when you put these two together, the overall energy of the reaction is 16.7 kilojoules per mole. And because it's a negative number, that means that the reaction proceeds forwards because it's exergonic. So we make ATP because it's a really good source of chemical energy, because energy is released when it's hydrolyzed, and we can team up that energy that's released with an endergonic energy requiring process. It's a lot more complicated than that. I'll talk about that more in Biochem 2, and hopefully you guys might think about joining me for Biochem 2 and I'll explain exactly why. But a big part, and now I want to talk about well, where does this energy come from in the first place? Okay, so you got we eat sugar, we go through the TCA, we go through glycolysis, TCA cycle, all of that stuff, and then we produce ATP at the end. What's happening in between? And this is, again, where the chemistry explains the biology. Because what we're talking about is the movement of electrons. And it's kind of framed by one of my favorite quotes, and I got it on my door in my office. Life is just an electron looking for a place to rest. And essentially what we're doing is that we are consuming electrons from a high energy source, a high energy state, like in carbohydrates or lipids. They're in a high energy state. and we make them move down into low energy states by the sequential metabolism of the intermediary metabolism. So as we go through glycolysis, we're taking electrons, we're taking them from a high energy state into a lower energy state, and then we keep going down, 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 down. Each step, we're pulling those electrons down into a lower energy level. Now that movement of electrons, that is, it's being oxidized. And you have the phrase oil rig. Do you guys do oil rig or Leo Ger? Some people do Leo Ger. I think that one's weird personally. Oil rig, right? Oil rig. Oxidation is loss, reduction is gain. We're talking about electrons. So the electrons are lost from one thing and they are transferred to another thing. And in chemistry, when electrons move, that does work. Movement of electrons does work. And think of it as being like an electrical circuit. And so life is really just this electrical circuit of movement of electrons. And that is why it says that life is just an electron looking for a place to rest. But it doesn't rest because it's always moving. We start out with a battery, positive and negative. Battery is an electrical circuit and it goes around. And let's put like a motor in the middle. And then we connect it up. Now, in electricity is actually the movement of electrons from the negative electrode, travels down through to the positive electrode. So actually, the electrical circuit goes from negative to positive. The electrons move from the negative, because electrons are negatively charged, from the negative, move to the positive. But as they move, they can do work. And so the work may be to turn a motor. But if I was to break the circuit, what's going to happen to the motor?
Stop working.
It's going to stop working. On the other hand, though, if I break the circuit here, what's going to happen to the motor? Because the electrons can still move and get to the motor. So I'm breaking the circuit after the motor or light bulb or whatever it is. Is it still going to turn? It won't continue. The light bulb will turn off. The motor will stop running. The electrons are going to there. So why is it not functioning anymore? Because they can't move. Because they can't return back. Because they can't move. It's the movement of electrons that does the work. And if they can travel through here and then they get to a breakage here, they can't move anymore. They get backed up, right? It's having a road to get to and from is all good. But if there's traffic on that road, no one's getting anywhere and no one's doing any work. You need to be able to get to your destination. And it's actually the traveling that does work. If there's a roadblock, you can't travel anymore. You just stop moving. If you stop moving, you're not doing work. So as sugar gets oxidized, it loses electrons because oxidation is loss. Oil, oxidation is loss. Your sugar that you consume loses electrons. At the same time, something is reduced. In glycolysis, TCA cycle, you guys are all biologists. What's the common thing that gets reduced in metabolism? There's a couple of them. Not yet. That kind of comes in at the end. But what happens? What's in between? NAD. So your sugar is oxidized and NAD is reduced to NADH. Oxidations are coupled to reductions. And that movement does work. And that work is generating the ATP that you then can use for other chemical reactions. We're going to talk about that a little bit more. But one thing I do want to really get across is that the more oxidizing potential something has, the more it can be oxidized, the more work it will do. And so the more it can be oxidized, if something is more reduced, it can be oxidized more. If something is less reduced, it can't be oxidized as much. So it's kind of think of it as being a battery. If something is reduced, very reduced, then it's a fully charged battery. If it's only half reduced, it's a half empty battery or half full battery, whether that's glass full or half glass empty. But so the more reduced something is, the more power it has in it. And so a really important concept that I really want to get across again And this lecture is like a lot of little concepts kind of stitched up together. A really important one is that fat has more energy in it than sugar. And people are often confused by that and surprised by that because you think of sugar as being the energy molecule. You think of you eat food so you can get your sugar up. It's something that we talk about loads. But fat has more energy in it than sugar. And the reason is that if we think about our carbohydrates, I think actually this is going to circulate for me. Our carbohydrates, well, it doesn't circulate. Our carbohydrates are going to kind of be characterized by OH groups. Oh no, it's not doing it. Okay, well, the carbohydrates are like this here. A carbon with an OH. This is a carbohydrate here. It's a super simplified version of a carbohydrate, but it's CO. It's an alcohol. It's an alcohol group that's kind of like a carbohydrate. On the other hand, if we think about lipids, it's going to be CH2. We've got these CH2s on it. And if we take it back even further, well, carbon dioxide. Now, think about what happens in metabolism. You consume sugar, and then it leaves your body as carbon dioxide. Now, the reason is that this carbon right here is fairly reduced. As a general rule of thumb, when something's got more oxygen attached to it and more bonds to that oxygen, it is more oxidized. When it has less oxygen and less bonds to oxygen, it's more reduced. And remember that a food source with more reduced carbon has more energy in it. So if we were to look at the carbon dioxide, this carbon is double bound to two oxygens. So it's highly oxidized. Whereas if we move across this way, this carbon has only got a single bond, one single bond to an oxygen. So it is less oxidized, it is more reduced. But this carbon has got no oxygen on it, so it's reduced even more. So the carbon in a lipid is more reduced than the carbon in the carbohydrate, because the presence of the oxygen in the carbohydrate slightly oxidizes that carbon. And if we were to go to a ketone or an alcohol, it's going to get more oxidized. But the carbon in CO2 is fully oxidized and has no energy left in it. And so that's why we excrete it. That's why we breathe out CO2. Because we've already used up all of the electrons in the carbon, fully oxidized it, so we just breathe it out as waste. Now plants are going to take that up and recharge it with energy from the sun, and then turn it into sugar by photosynthesis, but it's no use to us anymore. We're done with that carbon dioxide. It's fully oxidized. But if we take in sugar, we can sequentially pull off those electrons. And those pulling off electrons, what's that process called? Oxidation. You can pull off those electrons by oxidation, turn it into CO2, and then breathe it out. But there are more electrons in the carbon of a lipid because it hasn't got an oxygen already attached to it. So actually, when we eat sugar, we're eating a carbon that's already partially oxidized. Whereas when we eat a lipid, we're eating a carbon which is not partially oxidized. It is fully reduced, and so it has more electrons and a stronger grip on its electrons, so it has more to give to that NAD or FADH2, FAD to FADH2. So oxidation reactions release energy. A carbohydrate has less, the carbon in carbohydrate has less electrons, and so it has less energy. And the carbon in a lipid has more electrons, so it has more energy to give in a redox reaction when we break it down. So really, really key. I'm gonna say this like very clearly with a kind of like nudge and a wink for keeping in mind for the exam on Thursday, is that there is more. You will get more calories of energy from a sugar than you will from a lipid than you will from a sugar. You will get more calories of energy from a lipid than you will a sugar, because the carbon is more reduced in the lipid than it is in the sugar, because in the sugar it's got some oxygen bonds to it. In the lipid, it does not. So which is going to have more More energy, well, probably palmitic acid is going to have, which is a fatty acid, is going to have more energy than glucose because there's more energy in these hydrocarbons than there is in these alcohols. Because there's the electrons, this oxygen has already got some of those electrons, and so it's not going to give them up. So there's less to give to you as a being. The oxygen has stolen them instead. Whereas the lipid, there's no oxygen, there's no oxidation, it's fully charged, and you can grab all the energy from it. So that's a concept I really like to get across. And the numbers actually are that on average, a carbohydrate has 5 kilojoules of energy per mole, and a lipid has 9 kilojoules of energy per mole. On average, it changes a lot, and that comes up quite a lot in biochemistry. So I've already talked about this, I've already talked about this, and this as well. This is more reduced than this, so there is more energy in this carbohydrate. And so as we answered earlier, well, where do these electrons go? Oxidation is lost, so these are reduced, ready to be oxidized to CO2. If it's getting oxidized, it's losing electrons, so it's coupled to a reduction, something is gaining electrons. We answered earlier, what's the thing that's gaining electrons? NAD, FAD as well, but those two cofactors. And what do we do with them? Well, this is the structure of NAD. We take those electrons, we put them here. And what's NAD and NADH? It's just a carrier of electrons. It's just carrier, it's just a transporter. And all it's doing is that it just receives some electrons to give it to something else. It's just like a warehouse of electrons. It doesn't have a function. It's just literally just like a warehouse of electrons to hold on to those electrons. Interestingly, what do you notice about this structure? What family of molecules would you say that this structure belongs to that we've studied recently? Nucleotides. Nucleic acid is when you put together nucleotides, but it's a nucleotide, right? Because there's the base, there's the sugar, there's the phosphate, and actually the D in NAD stands for dinucleotide. So it's nicotinamide dinucleotide. That's what NAD stands for. And then this is the nicotinamide, the bit that's slightly different. So it's kind of like the base of the other nucleotide. And this structure right here, nicotinamide. Have you guys heard of nicotinamide before? Nicotine, yeah. It is structurally related to nicotine. It's also structurally related to niacin. You heard of niacin? Vitamin B3. So you say, well, why do you consume B vitamins? Consume B vitamins because they are cofactors for enzymes in energy metabolism. Vitamin B1 is required for pyruvate dehydrogenase to make, which is important in glycolysis to make energy. Vitamin B6, pyridoxal L-phosphate, is for making aminotransferases and breaking down amino acids. Folic acid is vitamin B5, I believe. I think folic acid is a B5. That's important for methylation reactions and transferring methyl groups between things, especially when breaking down your food into energy. Anyway, this one's niacin, vitamin B3, makes up NAD. And when it gets reduced here, reduced means grabs electrons, takes electrons. It has grabbed them by oxidizing a sugar. Those electrons go here, and it holds onto them. And then it can transfer them to somewhere else because its purpose is as a transferer of electrons or a carrier of electrons or like a warehouse of electrons in a roundabout way. And it's the same with FAD as well, but it gets reduced. And you see this, it has one hydrogen on there. When it gets reduced, it has 2 hydrogens, which means it's got an extra bond. And what is a covalent bond? A sharing of a pair of electrons. So by adding an extra pair of electrons, you add 2 electrons and a hydrogen to NAD, you get NADH. And now that NADH has got 2 electrons that it didn't have before. So it's gained electrons. So it has become reduced. And it can give away those electrons to become oxidized again. So NADH is useful in biology and biochemistry because its structure means it is easily reduced and oxidized, which means it can easily pick up electrons and then drop them off again. It's very useful intermediate in the transfer of electrons between your food and the processes that you want to do with that energy. And so the process will be that NAD takes electrons from your food, from your glucose, from your lipids, takes those electrons and is reduced to NADH, and then those electrons come off in order to do the chemical reactions of your body, in order to do those anabolic processes that I was talking about that require energy and decrease the entropy. You're going to transfer those electrons from NADH, restoring it back to NAD, where it can be reduced again, and it can just keep cycling around. So it's a really good redox molecule that's easily reduced and oxidized to pick up and drop off electrons, pick them up from your food, drop them off to wherever they're needed. And we're going to talk about one really good example in particular in a little bit. But if you don't get enough of NAD, you can, a common thing is a disease called pellagra, which causes dermatitis, diarrhea, and dementia. So these vitamins are really, really important. Or black tongue, which is a disease in dogs where they don't get enough niacin in their diet because they can't make NAD. And also, like I said, niacin is structurally related to nicotine and structurally related to nicotinamide, which is the nicotinamide in nicotinamide dinucleotide, which is NAD. So they're all kind of structurally related to each other, but don't mix up nicotine with niacin. They're slightly different. So that really covers the main part, actually, of what I wanted to talk about. So I've talked about, like, I've just defined like a whole bunch of like little terms. I've defined oxidation, reduction, the movement of electrons, the fact that your food is reduced and it has a high density of electrons. There's more electrons at a higher energy level in lipids than are in sugars, because sugars have that oxygen in it, and the oxygen is taking a share of the electrons, whereas that isn't present in lipids. There are more electrons in the carbon, but then that food gets oxidized and something else, and then doing so gets reduced, NAD or FAD to NADH or FADH2, which is like a holder of electrons that can readily give it to something else. And that movement of electrons is doing work exactly the same as an electrical circuit. So that's the kind of thing that I wanted to frame that. And the main one I wanted to frame is that I wanted to give the example of oxidative phosphorylation. And that's the last thing that I'm going to leave you guys with. And then we'll call it a day, and you won't have to listen to me lecture ever again. So unless you can join me for Biochem 2, and hopefully a few of you will. I know a few people are thinking about it, so that'll be really nice. Yes, I just wanted to frame that in the context of oxidative phosphorylation. So like I said, the metabolic pathways allow energy to be released from a variety of molecules. Like, you could just get your energy from sugar, but there's lipids present in the universe. So why don't you also break down lipids for energy? There's amino acids in the universe. So why don't we break down those as energy as well? Metabolism and all of the different metabolic pathways of intermediary metabolism allow you to get as much energy as possible from as many different processes as possible. But they tend to run over the same paths, because why have 10 enzymes when you can just have 2? If you just convert everything into the same thing, you don't need to have a separate pathway for it. You can just convert it into the same thing, makes it a lot easier to work with. Release of energy typically doesn't happen all at once. If you want to squeeze the most juice out of an orange, you just do it slowly, bit by bit by bit. Or if you have an orange, and if I grabbed an orange, I just squeezed it with my bare hands or put it in like a press, I wouldn't get so much juice out of it. But if I slice up that orange into lots of little slices and then squeeze it, I can get more juice out of it. Why are there 10 steps in glycolysis? You could do it in one, but you wouldn't get as much energy out. By spreading glycolysis into 10 different steps, you're cutting that glucose molecule into little chunks, and each chunk you can squeeze out a little bit more energy out of it. So why is metabolism so complex? Because by being complex, it can be more efficient. And you can squeeze every little drop of energy out. Glucose metabolism is one of the main sources of energy. We're not going to talk about glucose metabolism in Biochem 1, but I talk about it for about half of Biochem 2. But a breakdown of glucose will yield about 38. One glucose molecule will give you about 38 ATP molecules net. There's also you have to invest some as well, remember. But the net profit is going to be about 38 ATP molecules. And also, it also varies a little bit because some things go out and then come back in again, and it depends on which steps kind of go in and out at different places and whether or not you use isoform 1 or isoform 2, and it depends on the organism a little bit, but about 38 molecules for ATP. It could also, like I say, divert through other metabolic pathways, such as the pentose phosphate pathway, so which will shunt out and then shunt back in again. I'm not going to talk about the pentose phosphate pathway, but I do a lecture or two on it in Biochem 2. But it's just another thing that you can turn sugar into that is useful. And once you make ATP and producing equivalents is just chemist talk for thing, like NADH, because it's fully reduced, so it can reduce other things. You can make those, and you can use it and it can come from a variety of sources, and you can use it to build other things. And one of the key things is in moving around phosphate groups, such as ATP to ADP. That breaking that off, that releases energy, which is coupled to energy requiring reactions. But if we were to frame this all together, this is kind of what metabolism looks like. Sugar metabolism. Glucose comes in, it goes down through glycolysis from glycolysis that produces a certain amount of ATP and the NADH. I've colored in the NADH in purple because it comes in important later. But glycolysis produces some ATP and NADH. The rest of ATP goes into the TCA prep step, which is pyruvate dehydrogenase, where pyruvate is converted into acetyl-CoA. And that in turn generates CO2 and NADH. If you recall what I said from earlier, why do we produce CO2? We produce CO2 because we have fully reduced the carbon and we've got no use for it anymore. We've fully oxidized the carbon and we have no use for it anymore. The carbon had some oxygen bonds to it, and we made it more and more oxidized, pulled more and more electrons from it, and eventually had no more electrons to give. And it became CO2. And since it's got nothing else to give, we just breathe it out as waste. But in doing so, we also produce more NADH, and we feed that acetyl-CoA into the TCA cycle, and that will go around and produce more ATP or GTP, produce NADH, produce FADH2. But also, as we're doing that, we're oxidizing those carbons more and more and more, and producing 2 CO2 as well. There's actually 3 NADH, 1 FADH2, 3 CO2s, and a GTP. But GTP and ATP are readily interconverted. The point is that you oxidize it further, lose those carbons you don't need as CO2, and gain NADH and FADH2. So we've made a bunch of NADH. We haven't actually made very much ATP. We only make a small amount of ATP, only about 3. I've made 3 ATP up to now. We've made a whole bunch of NADH and FADH2. And what do we do with that? We feed it into oxidative phosphorylation. NADH, FADH2, and oxygen, which is why we breathe oxygen, feed into oxidative phosphorylation to produce ATP and water. And that's the ATP. That's where the rest of the ATP comes from. That's where the 30-something ATP come from. Up until now, you've only made about 3. The other 33-ish come from oxidative phosphorylation, come from feeding NADH into OXPHOS. But metabolism is super complicated. And like I said, you've got anaerobic respiration as well, where NADH, pyruvate is turned into lactic acid, and that will loop back again. But and this is kind of meant to be it will come out, but also go back in again. And so lactic acid is when you don't, anaerobic respiration is when you don't have oxygen. If you don't have oxygen, you can't do this. So there's no point doing this and this. So you just store it as kind of lactic acid for a little while, and your liver deals with it later in the Cori cycle, which we'll talk about in Biochem 2. You see where I'm going with this? So, glycolysis can divert out into anaerobic respiration. You also have beta oxidation, which actually would be better to feed into the TCA. It would make more sense feeding it into the TCA cycle rather than TCA prep. But really, I kind of put it there because it makes acetyl-CoA. And acetyl-CoA will feed into the TCA cycle, exactly the same as glycolysis feeds into the TCA cycle. So you break down lipids. You also break down amino acids. Amino acids can feed into the TCA cycle, but also you can pull out of the TCA cycle and pull out of glycolysis to make amino acids. So these are all complicated and interlinked. So all of metabolism, all these molecules we learned about amino acids, we learned about fatty acids, we learned about glucose. You can build them into complicated things or break them down into fuel in order to make ATP. And they're all interlinked because it's much more efficient to have amino acids and fatty acids feed into the sugar pathway than it is to have their own separate pathway. This makes it more efficient by reusing the same thing. It's like having, it's like carpooling. It's more efficient to carpool than it is for everyone to have their own car. That's the kind of the idea behind it. I'm going to skip over this slide, but this really it says what I've already said, is that we sequentially oxidize to get more energy, we compartmentalize to get more energy, but also by compartmentalizing, we can separate out hungry things from things that are not so hungry, so the things that are not so hungry get starved by the hungry things. If you've got a process that uses a lot of ATP, then that means there's not going to be much ATP for other things to use. So if you get that hungry process and push it off to the side and surround it with a lipid wall, the other things will get starved of ATP. So you have, like, let's say you've got something hungry, you put it in one room. You've got a bunch of things that are not so hungry, you put them in a separate room. By having them in different rooms, they don't interfere with each other and starve each other. So that's important. That's why we have lipids, to separate things out into different compartments. Metabolites can feed into each other as point 3. And point 4 is that this needs to be very tightly regulated, because you don't want to be producing energy when you're asleep, but you do want to be producing energy when you're running away from a saber-toothed tiger. So you've got to be able to switch metabolism on and off, because you don't want it to be on all the time. It's pointless having it on all the time. But you also need to be able to switch it on when you need it. So these are very highly regulated, and the enzymes are very highly regulated by that allosteric regulation that we were talking about before. So this is where it all feeds in. Actually, everything that we've done feeds into this, which is Biochem 2. This is how it all kind of fits in. I'm telling you guys about the molecules so you understand what we do with them and how we make energy with them. You guys all are all biologists. You know what a mitochondria is. Cytoplasm, outer membrane, inner membrane, intermembrane space, matrix. You guys know all of that. I'm going to go straight out over the slide. The chemists, I need to explain that to a couple of times. That's a lie, not a couple of times. Those chemists I need to explain that to, because they haven't done as much biology. But you guys are all biologists. You know all of this, so that's no problem. But the important thing is that these are lipid membranes. And these lipid membranes are separating things out. This is a separate compartment to this, which is a separate compartment to this, because they are separated by the lipid membrane. And that's what lipids do. They create different compartments. In the electron transport chain, you have 2 compartments. You have the matrix, which has few protons. And you have the intermembrane space, which has lots of protons. And they are separated by, in the middle, the inner membrane. That's what separates between those 2 compartments. And what happens in the electron transport chain is that you use the NADH and the FADH2. Those electrons that are on those molecules that have been holding there in that warehouse, holding on to them, they move. They move to the complexes. The electrons from NADH move to complex 1. The electrons from FADH2 move to complex 2. Remember what happens when I say when things move? What is that coupled to? What does it do? Sorry? No, that's oxidation. So the NADH and the FADH2 will become oxidized. The complexes will become reduced. But when electrons move, what does it do? Work. And the work is to pump the protons from one side to the other. Actually, what happens is that the electrons go to complex 1 and 2, and then they hop from 1 and 2 to 3, and then they hop from 3 to 4, then they hop from 4 to oxygen, which is why we breathe oxygen. But the work that's done is pumping protons to create a gradient. There is a gradient separated by a membrane. And I'm going to skip forward a couple of slides. That gradient kind of looks like this. Does anyone know what? I mean, I know that this is a dam. Does anyone know which one this is? The Hoover Dam. Yes, excellent. I always remember this one because the Hoover Dam is supposed to be really, really big and very American. And obviously, I'm not American, so I'm always just like, what are the American things, right? You know, I have an interest in the American things because they're exotic to me. So I'm like, oh, the Hoover Dam is this huge dam, very American. You know, like there's a bald eagle somewhere squawking over there or something. Apparently, you can see it from space or something like that? Can you? Like it's so big. No,
That's propaganda.
probably that's true. Yeah, this is it's the same kind of thing. You've got a lot of water on one side, and not very much water on the other side. And this water up here is potential. This is potential energy. And what do we do with that potential energy in a hydroelectric dam like this? Well, you're holding it back till you need it. That's one part of it. But what do you need it for? What does it do? Generates electricity. You convert it from potential energy into electrical energy. Right? Same kind of thing. The electron transport chain and oxidative phosphorylation is exactly the same thing. You pump a bunch of protons from one side to another. And because there is a lot on one side and not so much on the other, all of this up here, where does it want to go? It wants to go down. And it will just go down on its own. And as it goes down on its own, it moves. And so what happens, and I'm going to go back now to where I was before in the slides. Well, I mean, this is, you know, it goes down. And as it goes down, it does work and transforms from potential energy to electrical energy. Something that can be used, right? We can't use, in humans, we can't use falling water and a pool of water. We can't use that really for an awful lot of our tasks. We convert that into electrical energy, we can do anything with it as humans, right? We convert it into a more useful energy source. And so what's happening is that the complex 1, 2, 3, and 4 are pumping protons to create a chemical potential, to create a charge potential, right? Because there's a lot of positive charges here, and there's not very many positive charges here. So this is going to be positively charged, and this is going to be negatively charged. And those positive charges, they want to get down to those negative charges. And also, they want to, there's a large amount here, they want to get down here. So they want to travel back. They want to go back. So what you do is that you put electrons into complex 1. You put electrons into complex 2 via NADH and FADH2, respectively. And as they move, they pump protons. They transport them from the matrix into the intermembrane space, building up more here, less here. Right? This picture right here, I would get very familiar with this. There is less protons here. There are more protons here. And the protons are being pumped across into here. And these electrons, they start out in complex 1 and 2, and they move to complex 3, move to complex 4. I'm not going to go into detail about how they move and where they move and via what. That's all covered in Biochem 2. But they move from 1 and 2 to 3 and 4, and then eventually are accepted by oxygen. And as they move, they do work, pumping protons from somewhere that's deficient to somewhere where there's a lot of them. Because those protons don't want to be in this big field of the intermembrane space, because there's already a lot of protons. So you're going against the gradient. So you have to pump against the gradient. That requires energy. That energy comes from the movement of electrons that was derived from NADH and FADH2 that was derived from sugar and fat metabolism. And then at the end of the electrical circuit, complex 4, those electrons finally pass on to oxygen. Because remember what I said earlier about the electrical circuit. I see that from the Zoom video, you guys can't see this because this is really, really shaded. I wonder if, there you go. There's this electrical circuit going from negative to positive and going around until it hits a motor. If I break the electrical circuit here, the motor will stop because the electrons can't get to the motor. But if I was to break the electrical circuit here, the electrons can get to the motor, but they get stuck because there's a break here. It's not that, in fact, the electrons can get to the motor. It's being able to pass through and get to the end. And in order to pass through and get to the end, there has to be space to do so. So there has to be a final acceptor of electrons at the end. Because if there was not a final acceptor of electrons at the end, the electrons would go to complex 4 and then they'd stop. And then there'd be a block, there'd be a traffic, there'd be a block. The electrons can't go anywhere else. So they need to go to a final acceptor after complex 4. That final acceptor is oxygen, because oxygen really likes electrons. That's one thing that we know about chemistry. Oxygen really likes electrons. So it's very, very readily accepts those electrons from complex 4 and becomes water. And then that water could just leave our body or go to other processes. So electrons go in, they transport to complex 4. In doing so, pump protons from low to high gradient. And at the end, 2 electrons plus 2 protons plus half an oxygen. And it's half an oxygen because you can't have half an oxygen, but it happens twice. Produces water, H2O, at the end. And then that water can then just go and leave or get used in other things. And that's the reason why we breathe oxygen. We breathe oxygen to be a electron acceptor at the end of the electron transport chain. Because if we didn't, then there wouldn't be a final acceptor, and the electron transport chain would just stop running because there'd just be a traffic jam of electrons. They need to go somewhere at the end. So by attaching to oxygen, turning into water and leaving, then the traffic ends. There is no traffic. But if you didn't have that, then the traffic would get backed up. And if we didn't have oxygen, well, then if we weren't breathing, we wouldn't be getting oxygen. We wouldn't be producing energy from oxidative phosphorylation from that hydroelectric plant that we have in our mitochondria. And thus, we're not making energy. And like I said at the beginning, what happens if we don't have energy? We die. Because energy is required to keep us together as an organized being. And we would just break down and we would die, and we break down to ammonia and water and carbon dioxide. We would just rot and die because we have nothing to keep our shape in. But if we have energy, because we're breathing, we can keep our shape and stay alive. So that's essentially what's happening. And then finally at the end, those protons that we've built up, they travel down through ATP synthase. And as they travel down, they literally turn a propeller. And this is all actually summarized here. I would get very familiar with these 2 slides here. Maybe, let me think. About 8% of the points are on these 2 slides here. And literally, those protons that you've built up will travel down through ATP synthase. This is what ATP synthase looks like, and literally turn a little propeller. And ATP synthase is literally a little motor that turns. Literally, you all have little motors inside your body. They're called ATP synthase, which is in your mitochondria. And literally, as these hydrogens, as these protons travel through your ATP synthase, it spins a little motor like a turbine that generates ATP from ADP and PI. Literally, that's literally what happens. This slide as well is important. ATP synthase, protons flow down through it, turns ADP and PI into ATP. And for once, I'm actually finishing on time because these are the last 3 slides. I'm just going to summarize in these last 3 slides what this lecture was on. The energy transduction in living organisms follows the law of thermodynamics. So second law of thermodynamics, it costs energy to tidy your room. We all have a shape, we all have a form, our bodies are organized, our cells are organized, that requires energy. We don't put in that energy, we die, we rot, we turn into CO2. Brutal as that is. And so energy is required. Cells, and that energy comes from energy releasing reactions. And the classic energy releasing reaction is the cleavage, hydrolysis of ATP to ADP. That releases a ton of energy that we can harness to other reactions. Reduced substrates such as glucose, lipids being the more reduced, so there is more energy in a lipid than there is in a sugar. Reduced substrates are oxidized by many steps And those oxidations are pulling electrons off and putting them onto the cofactors that get reduced, NAD to NADH, which is the cofactor that's just holding on to some electrons that can drive the synthesis of ATP that we use for these things up here. There are many different metabolites in intermediary metabolism. Some of them are catabolic, some of them are anabolic, but they tend to converge. This stuff is not really super important. Neither is that top one. And in oxidative phosphorylation, electrons are transferred to complex 1 and 2 by NADH and FADH2, respectively. They then travel to complex 3 and 4, or eventually to complex 4, where it combines with oxygen to make water. And when they do, protons are pumped into the intermembrane space. This creates a reservoir of protons in the intermembrane space, which then travel down through ATP synthase to turn ADP plus PI into ATP, generating energy, literally turning it like a little propeller that we utilize. And that actually, that's everything. That is all I have to say about Biochemistry 1. Does anyone have any questions?
Do you have any, like, last minute, that's coming out?
My tips for the exam would be to look at last year's exam. There's up on Blackboard. Look at that. What I did was I broke it down. So I believe that last year I did maybe 30 questions. I think I've dropped it down to I've taken down 4. It's now down to 26. So I'm bringing down the questions, and I haven't really added in any harder questions. There's just 26 questions over covering starting out with lipids, moving on to carbohydrates, moving into nucleotides, and then finishing up with a couple of questions on this stuff. The first 2 subjects are probably the most heavily tested. The last 2 subjects are the least tested, but there's not an awful lot in it. It's like 60-40, but slightly less in the last 2 subjects. You look at last year's exam, a lot of questions, maybe a quarter of questions I just reused and I just jumbled them. And then maybe half the questions, about a quarter of questions I just jumbled. About half the questions I just said, a 6-membered sugar is called this last year. So this year I said, what's a 5-membered sugar, right? So you can, to a certain extent, you can question spot. But if you do so, make sure you read the question, because I have not copied and pasted any of them. They are all slightly different. And every year there'll be a question. And then last year it was the answer is B. And I'll ask the question again, but I'll jumble the answer and it'll be C this time, and people just circle B. That's because they just looked at the exam. It's just like, oh, it's the question. They just circle what it was from last time. You've got to actually look at the question. They do not do that. That happens every year, like lots of people do it. And also, like, for instance, don't memorize, 6-membered ring, pyranose. And then this year I ask 5-member, and you just go, pyranose. No, you've got to read the question, because it's none of the questions are exactly the same. But there are 4 fewer questions. There's no essay questions. There's the bonus points is at the back. The last page is a bonus points with 4 bonus points questions worth 3 points each. Make sure you look at those, because one of them in particular is very easy, and then the other 3 are a little bit harder. Some of the questions from last year's paper I just completely took out, and then I added questions from like other years. So I have all my exam papers, and then every year I take some questions out, put some questions in, jumble some questions around. So that's the way to do it. Make sure you can draw sugars. Make sure you can name fatty acids if I give you a fatty acid. Make sure you know the classes. Make sure you know your polysaccharides. I've never asked a question on polysaccharides until this year. And then there's, in fact, what I will say is the polysaccharide question. I'll tell you about the polysaccharide question. The polysaccharide question is, there's a table, and it says cellulose, glycogen, starch. Then there's a table, and there's a list of A, B, C, D, E, F, G, H. I've written a statement next to each letter. And then what you need to do is you need to say A, B, C.
Is it similar to the table that's in the carbohydrates slides?
Yes, very similar to the table in the carbohydrate slide. What to watch out for is that some letters may come up twice.
Oh. Yeah, we didn't hear that. I didn't hear that. You didn't hear that. I'm just nodding my head.
Some letters might come up twice. All letters will be used, but some of them are used more than once. But that's that question. I've never asked that question before, so I'm going to kind of see how you guys do, and then if you guys struggle with it, I'm going to change it for next year. You guys are the guinea pigs for this question. Sheila?
Okay, so for this lecture for today, I guess what you really went on heavily was the electron transport chain. Is that primarily what's going to be on there? Is there anything else from this lecture?
I think there's 4 questions. One big fat one on the electron transport chain, and then 3 others. And then of those 3 others, I'm not telling you. I'm not telling you what they're about. But there were a couple of times where I was just like, huh? There were a couple of times where I was kind of letting you know what was there. So if you were paying attention, you may have picked up on that one. But I think it's 4 questions, one big one on electron transport chain, 3 smaller ones on facts that I mentioned in this lecture. But like I said, some of the stuff I was just like, this isn't so important, and sometimes I was like, this is important. That's kind of a hint which ones are going to come up and which ones won't.
My question isn't about the exam, it's just in general. So when people are eating a keto diet,
yeah,
does the glucose pathway still get utilized at first, or does it turn into fat?
So with a ketogenic diet, what's happening is that you're not eating sugar, and instead you're eating so you're not eating carbohydrates, sugar. Instead, you're eating fats and proteins. Mm-hmm. And so what that does is that that produces an excess of acetyl-CoA. And that's because fats get broken down into acetyl-CoA, and several amino acids. So the amino acids are a mixture. Some produce glucose intermediates, but others produce acetyl-CoA. And what happens is you produce a ton of acetyl-CoA. And then that acetyl-CoA is then condensed. You get 2 of them, 2 acetyl-CoAs are condensed to an acetyl-CoA. I just did this today in the Biochemistry II lecture, actually. And they are condensed into an acetyl-CoA, which is a ketone body. And the point of the ketogenic diet is that it was invented in the 90s because as a treatment for childhood epilepsy. Because epilepsy requires heavy drugs to control it. But the problem is in a developing brain, that can damage the developing brain. And so it was causing some children to just be like catatonic and get learning difficulties because it's such powerful drugs. Like you can't just give a ton of diazepam to a 6-year-old. It just really makes them very sick. So they were like, well, we've got to find another way of doing it. What they found was that if you remove sugar from a person's diet, the child's diet, they stop having epileptic fits. And the reason is because you're producing this acetoacetate, this acetyl-CoA, and that is utilized by the neurons in your brain. And when the neurons switch to using this for a fuel, as opposed to lactate for a fuel, and both of these are supplied by the astrocytes So the astrocytes make acetyl acetyl CoA and lactate and feed it to the neurons. And but if you have a deficiency in sugar, you don't make lactate. You just feed them with the ketone body. And for some reason, when neurons use ketone bodies instead of lactate, you don't get epileptic fits. Don't know why, just do. People take it nowadays as a weight loss thing. And that is to do with just changing the metabolism in the body, and sugar is turned into fat in your body. And if you don't eat sugar, then you don't produce fat. So the big thing was that in the 80s, fat and lipids, fat is called fat, but also, and that just refers to the chemical lipid, fat. But also people call kind of cosmetically, they call kind of like deposits on your body as being fat. And so people, they're actually kind of two different entities. And people were thinking, oh, well, if I don't want so much fat on my body for aesthetic reasons, maybe I should eat less fat from the food. But actually they are not connected. If you eat a fatty steak, that fat doesn't just appear on your arm as like a lump of fat. It doesn't work like that. It doesn't, like the fat from food doesn't turn into fats on your body. They're actually two different things. They just unfortunately have the same name. And so what they did was in the 80s, there was a big push to be super skinny. So people were like, I don't want to eat fat, it'll make me fat. And so they were like, well, let's remove fat from food. But fat is flavor. So if you remove fat from food, you've got to replace the flavor. What do you replace it with? Sugar and salt. And salt is bad for your body. Sugar is bad for your body, but sugar, ironically, will turn into fat deposits on your body. So fat doesn't turn into fat deposits. Sugar turns into fat deposits. But by replacing the sugar or fat with the sugar, you get more fat deposits. So that's really the cause of like metabolic syndrome in modern day Western society, is because we've replaced fat with sugar, and sugar is more harmful than fat and turns into fat. So the ketogenic diet is not eating sugar, so that sugar doesn't get turned into fat on your body. And instead, in the ketogenic diet, because you don't have sugar, your body has to use other fuel forms. Those other fuel forms are proteins and fat, which decreases body mass and body size, because those are the things that deposit on your body. So that's what the ketogenic diet is. That was a very long answer to your simpler question. Does anyone have any more questions?
Well, you were going over the conjugate redox pairs. I think I didn't write down all that you said, but you said the more oxidizing potential something has, the more work has organized.
Yeah. So just if anyone needs to go, you're welcome to go. I'm just going to follow up these questions. It's like 3:20, 3:25. So the redox conjugates was to do with the idea that if we have we have a carbon, and this carbon is bound to a hydrogen, then it's a sharing of a pair of electrons. And those electrons they're going to be shared. And actually, carbon is slightly more hungry for electrons than hydrogen. So actually, it's got a slightly greater share of those electrons. Whereas on the other hand, and so this carbon is considered more reduced. Reduction is oxidation is loss, reduction is gain, but something is considered more reduced if it has more electrons or greater share of electrons. Because in this covalent bond, the carbon has a greater affinity for the electrons than hydrogen, has a slightly greater share of the electrons, so it's considered more reduced. On the other hand, you have an oxygen bound to it as well. This is also a covalent bond and a sharing of a pair of electrons, but that pair of electrons, oxygen, has a greater pull and affinity for electrons than carbon. So the share is not 50-50 in the middle, and it's not more over for the carbon. And in fact, whereas the difference between carbon and hydrogen is quite small, so it's kind of like a 60-40, it's actually more like kind of like 90-10. And the electrons are sitting more over on the oxygen side. And so this is a sharing of a pair of electrons, one from oxygen, one from carbon. Well, carbon used to have an electron, but it's lost it to oxygen. Because it's kind of partially lost it to oxygen, it is more oxidized. Whereas it's more reduced by hydrogen, because hydrogen's given it more electrons, more reduced by oxygen, because oxygen is taking electrons. And so it has become more oxidized. Or if we were to turn it into a double bond, well, now it's even more oxidized, because now carbon has lost 2 electrons, so it's even more oxidized. So as you go from a methylene to a alcohol to a carbonyl to 2 carbonyls for CO2, you are getting more oxidized. But if you were to give it electrons, it would get more reduced. And thus, a lipid is more reduced than a carbohydrate. And there is more energy in a lipid than there is in a carbohydrate because it has got more, it more readily gives away its electrons. It's got more electrons to give. Whereas here, this carbon's got 2 electrons to give. This carbon's got no electrons to give. Or if it was an OH, it would have 1 electron to give. So that's and it's giving it to typically NAD, NADH, NADFAD. Anyone else? No?
Great lecture.
Awesome.
A wonderful lecture.
Yeah.
Not about this, but so right now, like when people are digestive enzymes are trending, do they actually, help?